Okay, good morning to the first session, the first day of interspeech. Today is the session about disordered speech, and the first speaker will be Tanya Talkar from Harvard University. Please start.
Awesome. Hi, everyone. My name is Tanya Talkar. I'm from the Speech and Hearing Bioscience and Technology program at Harvard, as well as MIT Lincoln Laboratory. And in collaboration with individuals at the Walter Reed National Military Medical Center, I'd like to present our project, Acoustic Indicators of Speech Motor Coordination in Adults with and Without Traumatic Brain Injury. So to provide a little bit of background with regards to why we're interested in this, there have been almost 450,000 incidences of traumatic brain injuries, or TBIs, in between 2000 to 2020 for U.S. service members. And that's just counting service members. There are countless other civilians who also have TBIs. These individuals end up having a lot of long-term consequences of the TBI as they are recovering. But in particular, we're interested in the fact that these individuals present with a wide variety of speech impairments for which they undergo speech therapies as they are recovering. We believe that analysis of speech production in individuals with a history of moderate to severe TBI will help develop an understanding of the long-term effects of TBI, which will also potentially help develop quantitative measures that can track progress due to treatments as these individuals are recovering. There have been previous studies that have looked at using speech to monitor the effects of moderate to severe TBI as well as other TBIs. As part of the larger study that this project is a part of, it was found that speech during diadochokinetic tasks, or DDK tasks, was slower in adults with a history of moderate to severe TBI. Other studies from our lab have shown that correlation structures derived from acoustic features, which act as a proxy for speech-motor coordination, have shown promise in detecting changes in cognitive performance in preclinical TBI. And in addition, these features have also detected lingering subclinical impairments in mild TBI. So altogether, we believe that these measures of coordination that we can derive from very simple speech tasks can help detect and characterize the long-term effects of TBI on speech production. And I'll talk a little bit more in a bit about these exact features and how we plan to use them. So to very clearly state our goal, we want to understand, quantify, and characterize the residual effects of moderate to severe TBI on speech production. And we believe that these could be used to develop measures to track progress due to interventions or to inform the development of treatment trajectories that are personalized. So before I go into the actual methodology, I'd like to talk a little bit about speech production physiology and what we're working with when we are measuring speech motor coordination. So there are three fundamental subsystems in speech production depicted in this figure on the right: the articulatory subsystem, phonatory or laryngeal subsystem, and the respiratory subsystem. And so to actually produce speech that requires very complex neuromotor coordination within the subsystem itself. So for example, you need to finely coordinate the timing of the movements of your lips, tongue, jaw, and velum to be able to produce speech. But in addition to this coordination within each subsystem, you also need to have complex coordination across the different subsystems. So there's been research looking at the coordination of articulatory and laryngeal motions, the coordination of respiratory and laryngeal motions, and making sure that you need to have fine timings within each of these subsystems to produce correct speech. And so in the absence of us being able to actually measure the movement of the muscles within each subsystem, we use acoustic measures which we can derive from the audio of recordings of speech. So for example, for the articulatory subsystem, we can extract out formants to represent vocal tract resonances, which represent the movements within the vocal tract. In addition to these acoustic features, when we're measuring speech motor coordination, we can also use other features that try to get at a more physiologically accurate representation of the movements of the muscles. So one example of this that we used in this project were tract variables, which come from the KCE model from the Haskins laboratories. There has been a software developed at the University of Maryland under Dr. Carol S.B. Wilson, which extracts out 6 different tract variables that represent particular positions within the vocal tract during audio. And so these 6 variables are lip aperture, lip protrusion, tongue body constriction location and degree, and tongue tip constriction location and degree. These are depicted in the figure on the left. And so altogether, we believe that tracked variables can allow for a more physiologically accurate representation of the articulatory movements, and so we can compare them to the use of formants in our measures of speech motor coordination to further analyze coordination within the articulatory subsystem. And so altogether, using these measures, we believe that the coordination measures that we develop will show differences and highlight differences in complexity and precision of speech motor coordination in individuals who have a history of moderate to severe TBI as compared to controls who have no history of TBI. So diving into the methodology, we had 116 subjects that belong to a longer 15-year study that's being conducted at Walter Reed. There are 80 healthy controls that we analyzed. 37 of these are injured controls who had an injury in the past and are coming to the hospitals for further treatment, but there was no evidence of a TBI-related injury. And there were also 43 non-injured controls. The individuals with a history of TBI, there were 36 of them. 31% had a moderate TBI in the past, 38% had severe TBI, and 31% had a penetrating TBI. And these were all TBIs that had occurred in the past, and these individuals were recovering from them. All of the individuals underwent a speech protocol, which consisted of a diadochokinetic task, so the repetition of padaka, pa, ta, and ka. Each of these set of syllables were said individually as many times as they could in a single breath. They also read out the caterpillar passage, which was presented to them on a screen. And they watched a wordless picture story and were prompted to retell the story as part of a free speech prompt. From all of these audio recordings, we extracted out low-level features, which represented the different subsystems that we're interested in. So from the articulatory subsystem, we extracted out formants, F1 through F3, mel-frequency cepstral coefficients, and tracked variables, as I had talked about before. And from the laryngeal and respiratory subsystems, we extracted out fundamental frequency, speech envelope, harmonic noise ratio, cepstral peak prominence, as well as creak. And so all of these low-level features were used as time series from each of the recordings and were input into our algorithm to measure motor coordination. And so I'll walk you guys through this process of extracting out the measures of motor coordination that we use, using the example of frequency tracks of formants. They represent articulation-based vocal tract resonant trajectories. And so the first thing that we do is we can take the cross correlation of 2 of the signals. In this case, we're selecting F1 and F2 first. We then take that cross correlation output and sample it at a particular time delay scale. We have 4 different time delays representing different areas of interest that we're looking at. We can take that sampled output and use it to fill out one row in the sub-block of this larger matrix that you see. We then delay the signals relative to each other, once again sample the cross-correlation output, and do that 15 times to fill out that entire block. So the off-diagonal blocks are the cross-correlation across the different signals, and the diagonal blocks represent the autocorrelation of the signals. And so we can do this for all of the features that we've extracted, looking both within subsystems as well as across subsystems, and create these correlation matrices. From the correlation matrices, the measures that we're actually looking at are the eigenvalues of the matrices, which represent the dimensionality of the signals in a time delay embedded space. And so from the eigenvalues, we rank them from largest to smallest. We group them into the TBI group versus the control group, and we can look at the effect size of the eigenvalues from greatest to smallest. And so that's plotted in this figure. Let me see if I can pull out. It's plotted in this figure over here. So a positive effect size means that the eigenvalue is greater in the TBI group. A negative effect size means that the eigenvalue is greater in the control group. And this pattern over here in blue from positive to negative indicates that the signals in the TBI group are less complex or more coupled as compared to the signals in the control group. And the pattern in red going from negative to positive means that the signals are more complex in the TBI group and more random and erratic. And so altogether, the analysis of these patterns of eigenvalues derived from the raw time series using this correlation method can help characterize the nature of motor coordination in TBI as compared to the control group. So here's an example of a couple of the features that showed very prominent effect sizes and patterns as compared to the other features. And so we have fundamental frequency on the left and the interaction between fundamental frequency and formants on the right. So once again, the effect size, if it's positive, means that the eigenvalue is greater in the TBI group, and the eigenvalues are ranked from greatest to smallest. And so from these plots across all of the tasks, we can see that there's decreased complexity of fundamental frequency as well as the interaction of fundamental and frequency across all tasks for individuals who have a history of TBI. It suggests that these underlying signals have greater coupling of vocal source movements as well as higher coupling across the vocal source and articulator movements as compared to the control subjects. As I mentioned before, we were also interested in looking at the complexity of formants and tract variables and comparing the relative patterns within these two variables. And we found that for both of them, the eigenspectra indicate decreased complexity of articulator movements in individuals who have a history of TBI during the DDK and the read speech tasks. There wasn't much of a difference in the free speech tasks. And in particular, the effect sizes for formants and tract variables show very similar patterns, indicating that they both are representing similar movements within the articulatory subsystem and can be used for comparison. So in addition to characterization of the eigenvalues, we also use the eigenvalues in a Gaussian mixture model for classification between the two groups. We used an adapted Gaussian mixture model, which is used typically in speaker verification. And so we used the eigenvalues that were derived from a particular feature and task set. We then reduced the dimensionality of these eigenvalues using principal component analysis to extract out the top 6 principal components. Those features were then used to train a universal background model, which is the GMM containing 10 components over 4 training iterations. We then used the training set in a leave one out cross-validation scheme to adapt the model into a TBI model and a control model. And so the output score on the test subject was the ratio of the likelihood of the subject belonging to the TBI model versus the control model. The output score was then also used to fuse the outputs across different features and tasks to create a more robust model. So first, to calculate the fuse score across all features, we used the formula that's on the screen there. And we use a mask of the training AUC to be able to determine whether the score was included for that particular feature for the test subject. The scores across, fusing across all features was used to determine the fusion area under the curve within a task. And the scores were also summed across tasks to determine the fusion across all tasks and determine that AUC. And so the reason that we did this was because we wanted to combine insights from different features and different tasks to be able to contribute to a final, more robust model that would then help discriminate between the two groups. So for our classification results, we did find that fusing across all tasks and all features yielded an AUC of 0.78, which was greater than the AUC that we obtained on any individual task, though the DDK and read speech tasks did come relatively close with an AUC of 0.72 and 0.73 respectively. We also found that in comparing the classification performance of formants versus tract variables, that the performance of tract variables was equal to, if not greater than, the classification performance of formants. And so this indicates that there may be some additional information that we're able to get from the tract variables which aid in classification and are able to get at the articulatory subsystem. And this is similar to previous results that we've seen with the same comparison. So to sum up some of the contributions from this work, we worked with a simple speech protocol which has the potential to be used to assess the long-term effects of moderate to severe TBI. We also characterized motor coordination differences between individuals who have a history of moderate to severe TBI compared to controls with no history of TBI. We utilized measures of motor coordination that were derived from DDK, free speech, and read speech tasks in a fused machine learning model to predict the history of TBI in individuals. And we hope that we can use these features and measures to be able to track an individual's progress due to treatment or to suggest personalized treatment trajectories focusing on certain areas of speech production based off of the subsystems. We want to continue with working with this data set. There's quite a bit of data in here, so we'd like to further validate with a larger group of subjects, both within this study as well as other data sets that may be available. We'd like to look at longitudinal speech samples to see if the patterns persist or change as recovery continues, as that'll be important as we are trying to track these individuals through their recovery. We'd like to assess whether these patterns exist across different tasks, so counting or sustained vowels. We'd like to develop a better understanding of the link between information provided by tracked variables versus formants, and also assess the effects of other comorbidities such as PTSD, depression, mood disorders, and sleep disorders on both of these groups that we looked at. So thank you very much for your time.
Thank you. I have to sit down. Thank you. So, there seem to be no questions online yet. So, I open the floor for questions. If you have a question, please raise your hand. If you can ask the question at that chair there, then you will be visible for the online public. And we have a hand sanitizer for. So, any questions? Yes, Khalid. Please come. Yeah.
Is your eigenvalues, are they related to Lyapunov exponent?
Sorry, could you repeat that again?
The Lyapunov exponents, because here you are mapping your problem to, you are mapping the signals to the phase space using embedding, and then you are computing the eigenvalue of the integration matrix. So all this looks like computing the Lyapunov exponents, which is theoretically mathematically well known. It's a measure of global complexity. So do you have an idea about this? Or?
So I'm personally not very familiar with the concept, but can definitely look into it more.
Yeah.
We're trying to work with...
Because the problem of Lyapunov exponent is that when you don't have access to the velocity field, I mean, here I'm talking about mathematical principles, it's very difficult to estimate. It's very sensitive to sample size. And so I think it's worth to look at least empirically on the sensitivity to sample size, because there is actually no consensus. There is also condition of stationarity, and there is no consensus on how to compute robustly this exponent in practice.
Okay.
But I'm pretty sure that what you are computing is related to the Lyapunov exponent.
Okay.
But we can check it out.
Okay, I will definitely look. Thank you.
Okay, sorry, not enough hands. So we received some questions. I read it word by word. Hi, Tanya et al. I saw that you extracted other spectral and vocal quality features as well, such as CPP, click, etc., unless I'm mistaken. How did these features perform?
So the vocal quality features did not actually perform as well as some of the other fundamental frequency and formants. So we are looking into that a little bit more, but we believe that that may just indicate that these individuals do not have any differences with their vocal quality, and those performed less. Those didn't have as high of a classification performance as compared to some of the other features.
Okay, and we have more question from Nicholas Cummins. Do you think you could increase the accuracy of your approach by including more features reflective of changes in cognitive loading?
Yeah, so we definitely would like to explore a larger suite of the features and try and include those as well. They have been, included in different studies that we've looked at, and in particular, moving forward, if we could assess different features, we'd like to look at multimodal approaches as well. So trying to look at eye movements or handwriting and other fine motor tasks to be able to include that in addition to speech to make a very simple protocol.
Thank you. I think